Thank you all for coming. This is really a tremendous turnout. I'm Margaret Cianci, and I'm the founding executive director of the Alliance for Cancer Gene Therapy. And I really want to welcome you to our event today. So thank you so much for coming. This year, we celebrate the 15th anniversary of the mapping of the human genome. Not coincidentally, it's also that the first time grants were awarded to 3 young investigators from a newly focused organization, the Alliance for Cancer Gene Therapy, founded by Barbara and Edward Netter. Sadly, Edward passed away in 2011, but Barbara has continued to drive the organization forward. Barbara, thank you for all you do, and we'd like you to please stand so we may all recognize you. We've come a long way since the founding of ACGT in 2011. We have given away 55 grants, totaling over $28 million. And it's amazing what a small, passionate group can accomplish. Many grantees that ACGT funded early in their careers have gone on to be leaders in the industry, like Carl June. Many have also funded biotechs that were based on ACGT-funded research. And more importantly, now, there are 2 gene therapy treatments which have been approved by the FDA for particular forms of leukemia and lymphoma. We are now witnessing the golden age of gene therapy, or as some have said, the century of the biotech. But there is so much more to be done to make gene therapy a reality for treating all types of cancer. We know it can work. What we still need to know is how to make it work for solid and metastatic cancers, how to ramp up manufacturing, how we can treat more patients, and most importantly, how can we offer hope for all of those that are still fighting cancer today. And that's why we're here, to hear and learn about cutting-edge research being conducted by the pioneers in their field. I want to thank all of our panelists. Before I introduce our panel moderator, I'd like to thank our generous sponsors, the Barbara and Edward Netter Foundation, Guy Carpenter, Michael Gregory, the IHC Group, Penn Medicine, Pieris, Regeneron, thanks, George, Swan and Roy Thung, Eisner Amper, Selectus, thanks, Andre, Genève Corporation, the Lapin Foundation, Quarles and Brady, Quinnipiac University, and Robinson and Cole. I also take great pleasure in recognizing our chairman of the Scientific Advisory Council, Dr. Joseph Glorioso of the University of Pittsburgh. And I'd also like to recognize Dr. Bruce Levine of Penn Medicine and co-founder of Team Unity. Thanks, Bruce. I must thank Barbara Gallagher, Director of Philanthropy, for all of her dedication and diligence in bringing this event. And a heartfelt thank you to all of our volunteers and our board members for your tireless advocacy, devotion, and promotion of ACGT's mission. And if you'd like to learn more about ACGT as an organization, please take the folders home with you. Now for today's program. I am excited and honored to have Meg Terrell as our moderator. Meg is the biotech and pharma reporter for CNBC and appears on CNBC Business Day. She has covered new drug developments for cancer, compassionate drug use, pricing controversies, and chronicled the sequencing of her own genome, among many other topics. Meg, is there anything you haven't covered in the cancer space?
Viruses, as Joe told me before we started.
We are delighted to have Meg as our moderator today. And just in the back, there are seats if anybody would like to fill in. There's seats up here if you want to come forward and sit down. And it is really my pleasure to introduce Meg Terrell. Meg, thank you.
Thank you. So I'm super delighted to get to moderate this panel. I'll just give you a little bit of a roadmap about what to expect for the next hour, hour and a half. We're going to talk, I guess, for about 50 minutes, 55 minutes as a panel, and then we get to hear from Rachel, who has been through the CAR T treatment herself, and I can't wait to get to hear your story. After that, we want to hear from you. Think of questions. We'll have about 15 minutes for Q&A for everybody and Rachel after all of this. So start thinking about them now, as we'll get to do that at the end. So I'll introduce our panel now, starting with Dr. Carl June from Penn, really a pioneer, one of the pioneers of this technology. He's director of the Center for Immunotherapies at the University of Pennsylvania. Next to him, we have Andre Chulika, who's the CEO of Selectus, which is working on a next generation version of CAR T that would be off the shelf, and we'll let you tell us more about that. Next, we have who we're calling Dr. Oz, which I just learned is a nickname today. Oz Azam.
Either way will go, whatever you fancy.
All right. I hate when people tell me that, because then I'm like, I still don't know which one I'm supposed to say. Who is the president and CEO of Teamunity Therapeutics, which is a new company working on CAR T, and who previously, when we first met, was at Novartis, which brought the first CAR T medicine to the market. And then we have George Yancopoulos, who's the chief scientific officer and one of the founders of Regeneron, one of the biggest biotechnology companies in the world. And we have George's cast, who is also on stage with us. In this panel, we actually have 5 PhD and MD degrees, so 2 of those belonging to George, and 1, I guess, to his cast. So, Dr. June, let's start with you. Can you explain to us just what CAR T is?
So CAR T is in the end, it's a patient-specific therapy at this point made by your own white blood cells called T cells. And they're genetically modified so that they become cancer killers. And they can be given now in as few as 1 infusion. And our first patient was infused in July of 2010, and he still has CAR T cells. So it really sets it apart from all previous drugs, and that these are so-called living drugs and can act on long-term cancer immunosurveillance after infusion.
Mm-hmm. How did you get the idea for this?
Well, the idea, like a lot of things, there's a lot of fathers to it, and there's a lot of aspects that took decades to actually make work. So, the first idea of gene therapy happened in the 70s when DNA was cloned. So we had conferences then, because then people said, well, maybe you could change genes. But, it was thought that it would happen right away, and it took decades and decades to get FDA approval. Then one of our colleagues at the Weizmann Institute, Zelig Eshar, who was a basic immunologist, and that's an important aspect. He wasn't working on curing cancer. All he was trying to do is show how T cells that I explained, those are the ones we modify, differ from B cells. So in our body, B cells make antibodies, and T cells kill viruses, and they kill, they can see that a virus has infected a cell, and then they kill it. And he said, well, maybe we could make a hybrid, a Greek chimera, 2 species, B and a T cell fusion. And so he made what he called a chimeric T cell. He put the antibody from a B cell into a T cell and showed then that T cell could kill with whatever that antibody bound to. So it was a fundamental demonstration in the late 1980s. And then only later did they say, well, maybe this could be used to kill cancer or HIV. And so we began, Bruce Levine and I, in the 1990s, working with a small biotech called Cell Genesis to actually treat AIDS patients. Because back then, in the early 90s, there were no effective therapies for AIDS, and basically all people died actually because the T cells, the very T cells we want to use, were killed by the virus. And so a virus was made for another basic experiment at the University of California by Art Weiss and his colleagues, and they showed that you could now take the T cell, and if you put the antibody in with the signaling, the things that wake the T cell up and cause it to kill, it could kill and not only have the new specificity of the B cell, but also it made the T cell divide. And that was the genesis of the idea you could make a serial living drug, that one T cell could become two could become four, and so on. And so that was a demonstration we first tested in HIV patients in the second half of the 1990s. And we thought, okay, at that point, we did show in a randomized phase 2 trial that there was actually lower viruses, meaning the HIV in those patients. So it actually had some effect. But then pharma had a solution to HIV, which was in the late 1990s. They came out with very potent drugs, protease inhibitors, and it was no longer a problem. You could take a pill, and you didn't need an expensive cell therapy. So we had to abandon that. And then, but we got a real bonus because the FDA had a rule for all kinds of gene transfer therapy, which this was, because we used a new gene, put it into the patient's T cells. You had to follow those patients for every year for 15 years. And so our AIDS patients came back every year for 15 years. They went on the drugs that Abbott and others made that stopped the virus from replicating. And we looked and found out that a decade later, those patients still had their CAR cells, and that they had not essentially changed at all. They had a half-life of more than 17 years. So we said, whoa, this is not anything we expected. To be able to give a single infusion, and 17 years later, the patient still had them in essentially the same number that we infused.
Wow.
So we knew we could make a living drug, and then the same strategy was tried by two groups, one at the National Cancer Institute and one in the Netherlands, to try to kill cancer cells with these CARs, and they didn't work at all. They didn't last even for a week. So that lesson taught us that the cancer, what we call the cancer neighborhood or the tumor microenvironment, is much more toxic than someone who has a viral infection. So solid cancers kill T cells. And we had to find a solution for that in the field. About 3 labs, including mine and some others, another, Michelle Sadalan's here in Memorial Sloan Kettering, worked out a way to make the T cells so they could survive in a tumor microenvironment. We did that in around 2006. And we actually got funding from the Alliance for Cancer Gene Therapy then to do a trial to test that. And those CAR T cells then that had hardwired in proliferative signals that were resistant to being killed by the tumor now have survived, we now know, for 7 years. So there's many different incremental advances that I think is a lesson out of this. The idea never really changed. It's just we didn't have the toolbox initially.
Oz, why don't we go to you, and you can talk a little bit about Novartis and the applications there, what the earliest applications of this to getting to the market have actually been, what the opportunities have been so far.
Yeah, so I had the pleasure of working at Novartis for 10 years, and one of the highlights of my career there was to actually build out the business there in the cell and gene space. And so back in 2012, a unique partnership was struck between Penn and Novartis, whereby Novartis got exclusive rights to the platform technology. And then the real challenges start, right, in terms of taking what was a phenomenal set of initial patient results in a handful of patients that are incredibly powerful, dare I say curative, which was something a lot of people initially were very skeptical about. And that process was taken out of Bruce Levine's lab and underwent a process known as technical transfer to a manufacturing facility in New Jersey. And the process of then showing like-for-like comparability was done, and a scale-up of that process was undertaken over a period of time. And then in parallel, Penn continued to treat more patients under their IND direction. Eventually, Novartis then took over the regulatory reins, transitioned the know-how into the company, and then was very proud when my team was able to deliver a phenomenal result with one of the first CAR T approvals. And that, to Carl's point, I mean, what I've compressed into a couple of sentences, where it was built on the shoulders of giants, right? This was a 30-year process, starting from the early HIV work to then evolve and think to how to actually pick the right target, as George was talking earlier in the session, and honing this into a specific set of patients who would benefit, with the ambition that, starting with leukemia patients, lymphoma patients, we would have the potential to go wider. And that's a hope, I think, for many of us in the field, that how do we take this massive leapfrog now to the next level of technological thinking, now that we've mastered the fundamentals and the basics. And I will caution, it's still very early days for this field, right? I mean, this is generation 1.0, but it's an amazing 1.0 version that truly transforms lives and gives families and patients a lot of hope, at least in the leukemia and lymphoma space. And I think now the hope is, how do we now pivot and make these technologies more advanced? How do we make them more scalable? And how do we tackle the real difficult challenges that we have around solid tumors, which are going to require much more than the simple scientific elegance of a CAR T structure attacking an antigen marker and destroying the cancer. It's going to require a whole host of manipulations around the microenvironment, turning things on, turning things off, trafficking cells in a different way. So the challenges are there, but the progress of the science has been phenomenal. And as I say often, that if somebody had said to me in 2012, when I first got exposed to working with Carl and Bruce and thinking about how we were going to build out the first approvals, if somebody had said that going from 3 to 4 patients proof of concept data to an approval, I would have said it can't be done. But it happened within six years. And so that's a phenomenal compression of, some of us have been in this industry a long time and seen things take 13, 14, 15 years to mature from benchtop to an approved product. That was a miracle in itself. So how do we now capitalize on that and keep the momentum going and trying to treat wider patient groups? So just some thoughts there on my experiences.
Yeah, a lot to talk about how to bring this forward into more patients. I want to hear a little bit from Andre, just to give an introduction into what Selectus is working on and how it's different from the CAR Ts that were approved last year.
So what we work on is called off-the-shelf T cells or allogenic T cells. And the idea of the concept is try to get this to patient regardless if the patient had or didn't have T cells or had bad quality T cells or not. Just come with a product that would come from a healthy donor that have T cells in great shape, and that we will engineer and do gene therapy inside and gene editing also, in order to be able to make these T cells injected to the patient and break a paradigm. This paradigm is that T cells cannot be engrafted allogenically from a donor to a patient, because the T cell is usually a kind of a sniffer, kind of a foreign, like a police that would check the ID of all the cells. And if it checks something that is from the outside, it will do an alloreaction against it and attack it. So the cell cannot go from a donor to a receiver like this without removing the ability to recognize a foreign body. Therefore, we first take the T cell, rip off the ability of the T cell to recognize foreign bodies, which is the T cell receptor, by knocking out with gene editing the T cell receptor, like the chain, the alpha chain. And then after this, we add inside this a CAR. So the CAR would be the driver of the activity of the T cell. And the CAR has, obviously, through the engineering that has been done, through what Carl was describing, everything inside to recognize the cancer cell, or like the tumor-associated antigen, cancer cells are not, like the compartment where the cancer is present, to kill this cancer cell, and also to amplify. The only thing that gene editing adds, which is this kind of next generation CAR T, is, for example, making the cell compatible with standard of care, for example, chemotherapy treatment, a monoclonal antibody preconditioning, or, for example, suppressing certain pathways inside, such as checkpoint inhibition, etc. So the first patient that we have been treating had no T cells. Therefore, these patients were not, for example, responsive to what's called bispecific antibodies that take a T cell, drag the T cell up to the cancer cell and kill it. So like they were treated with a drug that was developed by Amgen called blinatumomab, and no response. And they had no response because blinatumomab need the T cell from the patient to make this anti-leukemic activity. And therefore, like to do a CAR T therapy, it was difficult because these patients had no T cells just to source the T cells and make the CAR T. And finally, they came up to us and asked us, could we use allogeneic T cells to try to dose these patients? And that's how like we started dosing the first patient, I think in June 2015, and close to 3 years ago. And since then, this approach of combining gene editing to CAR T therapy is moving forward, and I think that it's going to develop during the next years, because if the T cell is the real effector, the killer of the tumor, then we can continue to program this T cell to become more and more powerful in the future and more interesting. And the concept of allo also has another side, which these T cells don't stay forever in the patient. So maybe the response will be not as long-term as an autologous T cell therapy, potentially. Or, for example, would require maybe repeat dosing or doing like injection with different type of CAR T, etc. So it's a new development in this history that was described by Carl initially.
A lot to plumb there, and we will. George, I'd love to have your perspective just sort of regarding this, both from Regeneron's perspective, and does Regeneron have work going on in this space, or are you considering work going into this space? And also, as you've observed kind of the business of CAR T and personalized cancer therapies over time, of course, we know the Dendreon story, which was an early attempt to use a patient's own cells to treat their cancer, and unfortunately that company ended up going bankrupt. Is that a cautionary tale from a business perspective here? How do you look at that?
Well, there's a lot embedded in those questions. So first of all, everybody should know I'm a little bit of an outsider, as Meg sort of implied, because we are by no means involved in the building of this field like these guys were. And it's truly, I always use the word miracle, it's really a miraculous demonstration of going from basic science, mostly in the academic setting, to something that, as I said, is essentially almost curative. It's a remarkable story. And I should say, Margaret used the word, she described Carl, or she said something about, we have many people like Carl. I don't think there's many people like Carl. I mean, he's sort of one of a kind. But I think that the Dendreon story is not really relevant at all here, number one, because the problem there was, it was not that it was an expensive or it was a very custom and engineered solution, per patient. No, that wasn't the problem. The problem was it didn't really work. Okay, so here you have something that really works. So this is really, I think, the dawn of the golden age. I mean, this is the first real example of doing this sort of approach. We had a little pre-meeting. I likened it to the fact that before this, nobody even invented the spaceship. And these guys came up with the spaceship and made it to Mars. I mean, it's really miraculous. And it really, and we have people like Rachel and others who are living examples of this really works. This is not something that has just a modest potential here. This really can be game-changing. But that said, I think that as we also talked about in a little pre-meeting, and as Oz and Andreas sort of referred to, there's a long way to go. Because what everybody has to understand is what these guys really did is they took the cells in the body that normally help the body fight infection, and they put in a tumor GPS device. They put in a GPS device that finds and kills just the tumor cells into these killer T cells. And they very efficiently are wiping out the tumor cells based on this GPS. It's a great approach, and it's a great proof of principle. It's a great solution. But I think the real challenge is that the tumors that Carl and his colleagues very carefully chose to target here had a very specific target that the GPS easily recognized. Okay, the GPS was like it was a red light, and it was only on the tumor cells, and the GPS took the tumor cells right there. Most tumor cells in the body. It doesn't matter whether they're other kind of liquid tumors or solid tumors. They don't have such a very obvious specific way to target them. And that's going to be requiring the next leap in this technology, which people like Oz and Andreas and Carl himself, they're all trying to take it to the next level to figure out how they can really do that, both to make the process better and less custom and so forth, but secondly also, how do you now extend this incredible advance beyond the small numbers of tumors which it's applicable to. And just to finish your series of questions, in terms of Regeneron, yeah, of course, we are very excited. I mean, I don't think you can be, in the business now and not be doing things like this. Most of our efforts right now are in the more science and preclinical realm. And what we're putting all of our resources in are into this second generation challenge, which is coming up with the types of reagents that can allow you to target the tumor cells that right now are untargetable, or at least untargetable in a specific and safe way. And in parallel, I guess Andreas introduced this concept, we're also trying to come up with a non-gene therapy way of doing this by using what's called bispecifics coupled with T cells, which is a whole nother field, but it's very, very related to this, and in some ways, these are going hand in hand in providing proof of concept and direction for each other. So this is no Dendreon, okay? This is here, to stay. I think there's going to be enormous interest and investment both in the academic world and in the corporate world to try to take this to the next level. And even if it's a related solution, this, let's just say, I mean, we're dreaming that maybe we could do this with so-called bispecifics plus natural T cells or something like this. It doesn't matter in that this is the example that proves that you can redirect the immune system to kill and literally cure cancer out of the body.
So we've used the word cure a couple times now. And of course, the major question is, can you bring those advancements and that potential cure? It's hard even to say it as a reporter because you're so unused to saying it in the cancer realm. Can you bring this to more patients and potentially all patients with cancer? Where do the limitations of CAR-T end? And I guess that's a question for the whole panel, but Dr. June, maybe we'll start with you.
So, I initially could not even say the word cure, but now I can. I think, because if we talk to our first patients, and Bruce and I have kept in close contact, they're 7 years out, they consider themselves cured. That is important because psychologically, if you get up every day and still think, well, am I going to be taking chemotherapy again or whatever, that's a very different life, living with cancer rather than going past it. So I think for the first time we've seen in leukemias and lymphomas, patients can be cured. Now I'll also say back in the early 80s, bone marrow transplant was the first way you could actually cure some leukemias and lymphomas. And that basically installs, when you do the bone marrow transplant, someone else's T cells in you in a very similar way, but without genetic engineering, to what Andre was talking about. Use someone else's T cells, and they then stay on patrol, they can eradicate a cancer that chemotherapy was not able to do. So I do think we're all trying to get curative therapies. It takes time to actually prove that. What I can guarantee you that all of us have different roles in this is that we're all very persistent because everyone said it was impossible to do this.
Right.
Okay, and what unites all of us, I'm sure, up here is we didn't quit. We've kept going after this, and I think finally now there is enough of a belief in this that we have a real coming together of the resources, the industry, and the academic enterprise to actually make this happen. Probably one of the hardest things we're finding right now to launch this into solid cancers and all the rest is that this is disruption. What we're doing, right now medicine works on therapies that you give over and over again recursively without curing you, and so what happens if you actually give something, you give it once and basically you just need a follow-up every once a year? A lot of the medical system has to change, and we're going to find resistance to that. Our hospitals are built right now on a financial model of recursive therapy that doesn't work or has only minimal effects. So they want to have curative therapy, But right now, our reimbursement system doesn't do that. So there's initial science aspect and what we try to do, you know, in our academic early phase trials, but how do you move that into mainstream is very difficult, and it's going to take a lot longer than I think people think, especially in our country where we don't have a common healthcare system. So there are, you know, how do you move it into the community? And because if we had this and could actually cure all the cancers right now, we could not deliver it. There's manufacturing issues, and there's the issues that the medical field isn't trained. Now, in our analogy goes back, you know, to when bone marrow transplant was invented in the early 1980s, there were only 2 centers in the country that could do that. It was the Fred Hutchinson Cancer Center in Seattle, and on the East Coast, it was Johns Hopkins. And for a long time, the only place where transplants were done were there. These are bone marrow transplants, not like kidneys. And people said, literally, it will never happen on a general level. But over time, people learned how to do it, and maybe 5 years ago, you know, the millionth transplant was done. So it took time, and it took 30 years to really spread out. And I think our real unknown is how fast can we get beyond our bureaucratic barriers and the inertias and so on. I mean, look right now at disruptive therapies like taxi cabs versus Uber. And the difficulties there in launching something. And another thing we've learned is it's very much geographically distinct. So we were talking in our earlier session, I mean, the US has been quite welcoming to gene transfer therapy and CAR therapies. In Europe, I mean, and I just published a review in Science with some of my colleagues, we looked at geographic distribution of CAR T-cell therapies around the world. And the 2 leading areas are China and the US. There is none almost in Europe. And it's not because they don't have great science. It's because they're very you know, conservative and bureaucratic, and the rules are different than they are here in changing the status quo. So, you know, how you disrupt a system is different in our healthcare system than it would be, say, in a single payer system like Canada.
What about the scientific barriers to CAR T? Do you think it's just a matter of time that you find a way to apply this technology to all cancers or the majority of cancers, or are there certain places that this won't be the right therapeutic option?
So I am very much optimistic this can be done, but it's not going to be simple as in a CD19 CAR for leukemia where you're one and done. That's probably not going to happen again. So what I think will be required is combination therapies. It may require, you know, BiTE therapies like George is talking about, different, you know, be multiple targets. It won't be a single one. And it will also have to be cells that you can turn on or off. And that way you can manage, you know, to avoid critical organ toxicity and still have anti-tumor effects. So it will be combinations with other targeted therapies and checkpoint therapies, for instance. And there are many trials right now comparing and are giving combined CAR T cells with checkpoints like PD-1 antagonists or anti-CTLA-4, and those in mouse models are very synergistic. So we're going to see many combinations. I think the common theme will be this idea of synthetic biology, making things that go beyond what the natural immune system does. And with gene transfer technology, with gene editing technology, with recombinant, you know, protein engineering, we have amazing possibilities now that get beyond what the natural immune system never was able to do.
And Oz, you're pursuing some new targets at Team Unity.
So I think it's beyond just the targets, as Carl's saying. So, you know, targets are one thing, but it's understanding the whole microenvironment, that ecosystem around the tumor, right? And I think that learning has increased exponentially in the past 2 decades, right? If you look at, you know, genomic tools and technologies, our understanding of cancer biology, there's a lot we don't know, but there's a lot we do know from before. So I think companies like ours are not just looking at that pure action of a CAR T or a TCR attacking a specific marker on a tumor cell. It's beyond that. So it's thinking about, well, how do you traffic that cell better? How do you, influence a promoter of that tumor in the environment? Can you switch that off? Can you put something in the cell that packs a punch and can, as well as destroying the cell, make sure that the environmental factors are manipulated and controlled? that's the tough bit. I mean, cancer's a fickle foe, right? You have mutations, you have tumors that escape. So to Carl's point earlier, I mean, I think that there will be, you know, certainly the hematological malignancies. We can now, I think, see that there is a potential that we can look back in 10 years and say, hey, you know, we cracked certain forms of leukemias, acute lymphoblastic leukemia being one of them. I think we'll have a really good inroad into treating lymphomas. The data's emerging stronger than before, than ever. And I think conditions like myeloma, if you think about those being a mixture of liquid-solid presentations, there's certainly hope there. Now, whether we'll get to, you know, lung cancer, ovarian cancer, pancreatic cancer, I don't know. But I think a lot of people are going to give it their damnedest to try to do it. And I was reading the other night a great book. I think many of you probably read Sid Mukherjee's book, The Emperor of Maladies. And there's one piece that always sticks in my mind where he talks about Dana-Farber in 1943 doing the first trial in children with aminopterin. And there was hope and there was glimmer, and he described it so vividly. But then all those children relapsed and unfortunately died. Fast forward 70 years on, though, right? You look at the papers that Carl and others published back in 2012, and now we have that hope of cure for that population. That took 70 years in the making, right? And you think of all the medical investments that happened and all the scientific parallel tracks that have happened. And I look back at what's happened in the past 20 years from a genomic sequencing perspective, the work on gene editing. You then couple that with our real understanding of manufacturing science and enablement. And so you bring all of that and accelerate it together, and I think there is hope that certain tumor types, for example, some areas I'm very optimistic about is glioblastomas. So you start to look at the fact that if, you know, a few years ago people never thought T cells would cross the blood-brain barrier and actually traffic to a tumor. Carl and team showed that that can happen. So if you can get that much happening for a T cell in certain tumors like glioblastoma in the brain, then what's the next level of evolution you need? And I don't think we're that far off on certain tumor types like that to be able to show a response. Now, whether we're going to be successful with, you know, the big cancers, that remains to be determined. But I think certainly the foothold is strong, the foundations are now stronger than they ever were. And I think it's going to require a lot of investment. I think it's going to require a lot of synergies between different players. I mean, the regulators are going to be key here. And in the US, I think we've got a very fertile and receptive audience, but I hope that that pollinates across the world, right? Because we want to push these therapies as quickly as we can to all parts of the world. So just my own personal thoughts.
What is your sense, and this is a question to the panel, of the enrollment of patients into clinical trials? Do you have trouble finding patients to enroll in the trials? I think ACGT gave me a stat, something like only 4% of cancer patients enroll in clinical trials, which sounds very small. What are the barriers to that?
I mean, there's start and kick off. I mean, you know, certainly in the cancer space, I mean, speaking from a company perspective, so the challenges will be that, you know, we're a highly regulated industry, right? Even our academic partners are highly regulated, and rightly so, right? I mean, there's a reason those regulations are in place. So being mindful of that, most trials require a minimum certain amount of criteria for patients to enter. And it's a real art between developing those studies and protocols to allow the right populations to enter, to ensure that you're actually studying a population where you can show a benefit and show safety. It's that benefit-risk equation. If you open to all comers, then sometimes that's not going to be the smartest move in terms of showing a particular therapy works. But there again, you have patients that are knocking down the door to get into trial. So it's getting that fine balance between getting patients in that express a certain tumor type, have the right biomarkers, have the right expression of the tumor. And that takes time, and I think that's when you're looking at those kind of numbers. Part of it is the challenge of finding those populations and screening out patients that maybe are not going to respond because they may have, you know, leukemia, but they may not have a CD19 leukemia, for example. So that process of attrition and screening takes out a lot of patients, but there's a reason why that happens, because, you know, we've got to work in a way where we're targeting these trials to actually benefit patients that are really going to truly benefit, and not harm patients, right, that won't respond. And let's not forget, the first generation of therapies in the CAR T space, these are really, really powerful therapies. I mean, T cells pack a punch, and you get major side effects. You know, you get things like cytokine release when you know, the tumor is being devoured by these T cells. You know, toxins are released, proteins are released, and this can be really detrimental. Patients can die. So these are not therapies for the faint-hearted, right? They require a lot of support, administration, and the right physicians who know how to manage these patients, right? That's the first generation now. Clearly, our understanding of safety and all these things are improving. So you have to think very carefully when conducting these trials. Okay, are we picking the best population of patients? Are they going to be best served with these platforms and technologies? And how do we ensure the right criteria where we don't lock out too many patients, but we get the right patients in? So it's a very difficult process.
Do you
But I
feel
think
like we
others
have
have
the
a lot
right
of
balance
experience.
now, or do you think there needs to be changes, and if other people want to jump in too?
I think the balance is improved. I mean, I certainly think the more dialogue we have with regulators and we're able to debate with them and discuss, you know, what, here's the protocol, let's see how you can help us relax certain criteria because we have the evidence for this, then, yeah, certainly you can make inroads, but it's not an easy business. But I'll give the other clinicians a chance who have a lot of experience on the academic center side as well as to some of the challenges.
Yeah, I'll just say, I mean, it's a very, very difficult question that's very sophisticated, though there are the issues of which the FDA wants, which is, I mean, they tell us who we can put on our trials, and some of those times we disagree, and other times we completely agree. And they're trying to make it as efficient as possible. I mean, and then there is this, it seems, slow pace of going from an early treating 3 patients to getting FDA approval. So I can't tell you how many emails I get from patients who want to be treated, and we can't because, I mean, right now, I'll tell you an example. We have a trial in prostate cancer. We treated the first 3 patients in a cohort in November, December. Then we have to wait six weeks before we can go to the next and treat patient 4. And I got an email today from someone in UCLA who wants to come and be treated. I said, no, we can't, because we have to go through this process. It's to ensure safety. And unfortunately, there's examples where in the past there were corners cut, and patient safety was compromised. So that's the highest ethic. But on the other hand, I mean, there's a big emerging political issue on right to try. And I can tell you, if I had cancer, I would want to try. You know, so what I have found is I've had you know, people who self-refer, and I've had several Nobel laureates who have cancer say they want to be treated. And it's very interesting. What I've learned is that people have an appetite for experimental cancer therapies that's independent of IQ and training. There are some, no, and I mean this, so there are some people who, very sophisticated thing, they basically, if they get cancer, they say, well, it's time to go fishing and die. And they really don't care and want to go, they figure this is it. And there are others who say, even if it's a 1 in a million, I want that home run. And what I found is that's independent of education level. You'll find people who only drove a taxi cab, and yet they will be to get to the front of the line, and they want that experimental therapy. And there are others who are you know, just won't do that. So I found that it's an inborn personality thing, this risk taking.
Yeah.
Okay, so how do you then tell someone who wants to take a risk that they're not allowed to because of these rules? They would rather say, and so we protect them and perhaps overprotect them by not allowing them to be on these trials. So there is, right now we do it at a population level, but it's amazing if you would go through the people in this room and give them the same scenario. Some would say, no, I don't want this. And others would say yes. And even if they're scientists and understand at a very high level, they'll come to a very different conclusion. So that's one part. And then you have the regulations. The FDA is trying to do things that are very innovative, like basket trials. So right now we have this approval process that we all have to work with, which is the FDA approves trials based on pathologic diagnosis achieved 50 years ago by what the cell looked like under a microscope. So we call, pancreatic cancer is this, and a lung cancer looks like this. Well, it turns out that they're really better classified by what the molecular mistake is in that tumor. And then, so like there is a gene that goes haywire called KRAS. and that's in some of the worst pancreatic cancers and colon cancers. And likely they respond in many cases to the same kind of therapy. And whereas other colon cancers don't have KRAS, that therapy would be not helpful. So one idea is this basket idea where you take tumors that are different, could be a lung cancer in one case, colon in the other, but they have the same molecular target, and then you could treat them on the same trial, and the FDA could approve that at the basket level. So far, they haven't done that. You know, they're all like, we're going one by one in the blood cancers, from acute lymphoblastic lymphoma to mantle cell, chronic lymphocytic leukemia, and so those are all B-cell tumors. They all have CD19. So anyway, there's a disruption happening there on how you do the trials, and it's complex on who to let on in many different ways. And it's medical ethics, it's a scientific part, and then it's the feasibility of maybe you can't even make enough of these cells at this point to treat 100 patients. And then how do you know, it's like liver transplants where there's many more people who need a liver transplant than we have livers. So you have to have a prioritization, and unfortunately it's rationing.
Well, I want to talk about safety. It's come up a couple times. Of course, in the development programs that we've seen, we've even seen clinical trial holds. Juno, of course, had some deaths in a trial and were put on hold and then fairly swiftly taken off that hold by the FDA. What would you guys say about where our understanding stands as to this safety profile of these treatments and how well it can be managed?
Well, maybe I can answer this on our side, because we started last year a trial on a new target, which was called CD123, and this target is expressed on myeloid progenitors and also on multiple myeloma cells, not on acute myeloid leukemia cells, also on another cancer called BPDCN, which is a more complex name. It's a very rare form of cancer, and this target CD123 is very well expressed there. And when you start a new therapy, you have to guess, it's kind of a poke in the dark exactly what kind of dose you're going to give, what are going to be the side effects. Are you going to, for example, totally wipe out the bone marrow with the product that you're going to give, or only kill the myeloid progenitors, the cells that generates probably the leukemia itself, etc. So these questions are unanswered, and any kind of animal studies that you're going to do will not answer this question. The only way to answer this question is to inject the product into humans.
That's for sure.
It's the real clinical research itself. So we started injecting the first patient at Cornell University that had an acute myeloid leukemia with a pretty high tumor burden, and at a dose that was 600,000 cells per kilo approximately. At our surprise, we thought that the product was at a very low dose, and it seemed to be at a dose that was therapeutically significant. So to our big surprise. The second patient was done at MD Anderson, and to answer your first question, by the way, you have very limited number of trials for the simple reason it's not because there is a lot of people that would like to get onto trial because they don't have another solution, and sometimes they relapse from a drug and it's the last line of drug, and they would like to get on new experimental drugs, but only certain centers such as UPenn, for example, MD Anderson and other centers in the United States can be prepared to handle such type of very experimental therapies that are sometimes also double-edged swords with cytokine release storm. Sometimes you can ablate also an organ, or it could be really risky. And the second patient that was those at MD Anderson that are very well trained had pretty poor prognosis for his BPDCN and was injected with UCAR123 and went into a grade 4 CRS and died. Unfortunately, he had a collapse of, he totally lost blood pressure and died. And we had a clinical hold from the FDA, and interestingly, discussing with the FDA of what happened, there was of course an investigation. There was a very in-depth investigation of what happened there. You can have a very constructive discussion with the FDA to try to resume the trial that took us 2 months up to the time we got there, and we had to de-escalate and re-escalate again the dose to try to find what is the right therapeutic window. So if there is, of course, like risk assessed to any new drug that you inject to patient, because, for example, if you use a monoclonal antibody that have a very good therapeutic effect or correct on certain type of cancer, a monoclonal antibody would work on cells that, for example, express over 100,000 cells of 100,000 cells of molecules on the surface of a cancer cell. Under 100,000 molecules on the surface of a cell, then it's not going to work as well. So you have certain threshold. For a CAR T, it's way more powerful. You need, I don't know how many, probably maybe less than 100. And so like the threshold of detection of a CAR T is way better and can go on a what's called an on-target but off-tumor that could ablate, for example, a healthy tissue. And that's why I think Carl was saying maybe you should be able to switch off the CAR T at a time to have less harmful effect. And the discussion that you have also with the FDA helps also to try to tune this up and bring probably these very experimental therapy to the right place, which is not very easy at start. And like these holds and resume the trial are probably a healthy system.
Carl, do you want to talk about just the therapies that we have on the market and the understanding that we have now of how to manage the safety of those therapies?
Yeah, so the 2 approvals in the US and still no other commercial approval outside the US, it'll be in Europe later, but for both Kite and Novartis are for acute lymphoblastic lymphoma, or leukemia, it's also called ALL, age 3 to 25, in people who have failed or can't go in remission with chemotherapy. And now diffuse large B-cell lymphoma by Kite, which is, and also again, it's for people who have failed. So, and that's a
or
population.
The therapies have failed them.
Yeah, they have failed at least an autologous transplant or more than 1 chemotherapy. And those are people of all ages, and I think it's up to age 70. I'm not sure if they made an upper limit. So diffuse large B-cell lymphoma is about 40 or 50,000 cases a year. There's others in the US, a total of maybe 100,000 a year of B-cell tumors. And so those are FDA approved. The manufacturing is being built out as we speak. Different centers, it's about 30 centers that Novartis has now that have been qualified. You have to learn how to obtain the cells in the blood bank, and then the patients, the physicians have to be trained on managing cytokine release syndrome. It's very unusual in that the CARs were approved for both Novartis and Kite with the drug called tocilizumab, which treats the cytokine release syndrome. So it's called a co-labeling. It has a black label for 2 indications. One is it causes cytokine release syndrome, which can be a fever. And initially, it always starts with a fever. You're going to hear from our first patient, Miss Elliott, who had, we'll see what hers is like. Our first adult had a fever of 107 degrees. And when the nurse saw it, she threw the thermometer away and said it was broken, and went and got another one and did it again. And again, it came out 107. So they had never, ever seen that before. That's a synthetically hyperactivated immune system. And it was when the tumor was being killed. So the physicians have to be trained, and the nursing staff, how to manage this. Now we had, initially there were fatalities, but in the trial that Novartis did, and this was international, there was a 48% incidence of grade 3 and 4, which means high-grade fever, probably ICU care, but not 1 patient died. So half of them get it, but they all get over it. It's completely reversible. There's another side effect, which has a black label warning, which is neurologic toxicity. And that also is reversible, and we don't know the cause of it. So we know the cause of the cytokine release syndrome. It's the activated T cells killing tumor. So it actually doesn't happen in the patients who aren't responding. So it's a one-time in oncology, when people are having a side effect, they start smiling, literally. So the neurologic can be an issue. There's one extreme, which is cerebral edema, which has really just happened in the Juno trial. No one really knows what that is, and it's rare, but it's being studied. One principle out of this that basically Andre mentioned is, in all of oncology, you can find out in preclinical mouse models does it look like it has promise, but we never really find the toxicities until we do human trials. So unfortunately, we need humans to volunteer, and that's, you know, when you discover, are there unanticipated toxicities? And there was with the CAR T cells, but they have been manageable, and that's where we're at.
I want to talk a little bit about price. George, when you saw the price of the first CAR T in the US at $475,000 from Novartis, Kymriah, what did you think?
I don't know why you would ask me the price question.
Yes.
But I do think that, you know, I have obviously a lot of different views about price. But, and one
Which
one
is why
way,
I asked you the price question.
one way that people come up with pricing solutions, or how you come up with it, for example, people who study this, like the Institute for Cost Effectiveness or ICER and so forth, is the value that's given to the patient. And I do think that it's defensible, and I don't know if ICER did an analysis here, but if you are actually saving a life with a one-time treatment, I think it would be defensible to have a very substantial price for it. The other part, though, that this value-based approach does not integrate into it is not only the value to the individual patient, but the value to society of having brought this forward. Okay, because as we've all talked about, this is just a stepping stone. This is just the beginning of the whole next possible generation of approaches. So society should not just be looking at this as the value per individual patient that's actually being treated, but also the value to society of bringing a new foundation that we can then stand on to go to the next step. And I think that as a society, with all this mania about, you know, high drug prices and universal health care, and everybody deserves health care, we're losing sight of something. That the most important thing, one of the most important things, arguably, to our society, is to come up with new therapeutic solutions for where nothing exists. And when one brings such an advance forward, it should not just be valued and priced for the benefit to the individual patient, which in this case is actually substantial, but also there's enormous value. If this becomes the stepping stone that does indeed come up with cures for all sorts of other cancers and so forth, you know, society's got to recognize that value. And we should be incentivizing the scientists of the future to be willing to take the risks, but there's also an enormous amount of financial risks to bring these forward. So that sort of value's got to be integrated into it. So I think that the price is easily defensible, not only on the actual value delivered to the individual patient, but on the potential value of this kind of innovation overall to society.
There's a real conundrum here, and I hear both sides of it. In some ways, that this is $475,000. How can any patient or, you know, healthcare system afford this? At the other side of that, some people question whether Novartis or Gilead, with its treatment that it acquired with Kite, which it priced at $373,000, will they even break even at those prices? And you also compare the gene therapy that was approved last year for a rare form of blindness that was priced around $850,000 for that one-time treatment for an eye disease. So these are very different price tags. How do you think about that?
So look, I can't comment for Novartis or Gilead or other companies, right? But I think don't lose sight of the fact that these therapies are the first generation and they are really expensive to manufacture. And I think that's a key point to bear in mind in that equation, right? Which is why now the next evolution of these therapies is really going to be not just on the science around the targets, the microenvironments, how do you scale these products and make them cost effective? Because the cost of goods for these products are high. Mm-hmm. And that's just the reality of the business. It's where biologics were 30 years ago. You know, you talk to people who were in the heyday of developing early biologics, and you know, large investments were required at that time. People balked at biologics, and look at biologics now, right? They're relatively affordable. And I think this is just the start of the journey for precision medicine therapies like this. So a big part of the conundrum for a lot of the companies, including my own, is going to be how do you now move the needle not just on the science, but in parallel, make the investments you need early on in the manufacturing sciences area, and that's something Andre and his company have invested a lot of time and effort early on as well. Because if you don't master that, then you are going to have consequences for the healthcare system downstream and a knock-on effect.
Can you describe, and Andre, certainly.
Maybe one thing I'd like to say, like how we got into the CAR T space initially, like back in 2010, like our CSO went to a conference and he saw like a talk of Carl June and Michel Sadelain, and he came back and for the first time explained to me what were CAR T. I got to listen to him very carefully. I said, wow, this looks spectacular. It was super exciting. And I told him, well, this looks like a procedure for hospital. And these are like doctors at hospitals. It's like a bone marrow transplant. It's normal. They do the apheresis. They know how to do GMP viruses. They know how to mix them with CAR T cells. And they can re-inject. And this can be done totally inclusive inside hospitals. We're a private company. We have to work on trying to do something that would be an off-the-shelf where the price should be valuable for the company and also for reimbursement, etc., up to the time where it can come with reducing the cost of good and having like several vials, what's called an off-the-shelf therapy. But the real milestone behind this, and I think we shouldn't look only at the price today. These first gene therapy and CAR T therapy that are coming to, like it was a big like milestone, 2017, in a year of gene therapy. It's unbelievable. 3 therapies were accepted by the FDA, notwithstanding what the price is. And this can only improve in the future. So don't look at this now. Look at these therapies that have been approved and how many trials, close to 300 trials worldwide on this. There is a big wave coming up. And these approaches that I think were academic and transfer after this to Novartis and after this to Novartis from UPenn and to Kite from the NIH. Now it's coming from companies such as Immunity, such as Selectis, etc., which has more like an industrial approach.
But I really do think that, as a healthcare industry, because some companies have made a lot of mistakes and have done the wrong thing in terms of drug pricing, it sort of captured us all into this morass.
That's right.
And everybody's always apologizing about pricing, okay? And people are talking about the cost of goods. It's justified based on the cost of goods or the value to the individual patient. And we got to get out of that mindset. Otherwise, our economy is going to not come up with solutions to the biggest problems facing our economy, like Alzheimer's, like type 2 diabetes, let alone cancer and so forth and so on, unless we reward innovation. And what we have to say, there's a term in economics called excess profits, which means too much profit for the amount of investment you put in. Okay, do you know what the #1 industry is? I would argue, being a citizen of the world, that excess profits should be highest in the things that are challenging the most important aspects of mankind's survival, like climate change or like healthcare. Okay, do you know where excess profits are highest now? Which industries? The tech industry, Facebook, Google, and so forth. Their excess profits on what they call innovation dwarf that in the healthcare industry. And you know who's lowest on the totem pole in the whole healthcare industry? The drug developers. Actually, it's the insurers and the middlemen, the PBMs, that have the highest excess profits. And the people who are developing the new drugs are lowest men on the totem pole. Yet we're the ones who are always being highlighted and said, oh, how you justify this sort of price? I would argue that excess profits should be highest to incentivize the next Carl June and the next geniuses here to sort of say, okay, we're going to do this. Yeah, because we're totally motivated to help patients and all that. But there's also going to be an enormous time and financial risk that goes into it, and we're willing to make it because the reward here can also be great. And I think as a society, we have to start recognizing and we have to start realizing that if we don't reward innovation in the areas that we most want, I'm sorry guys, but I don't need a new kind of iPhone. Okay? I don't need a new social media app. I don't think that's doing anything for us. Okay, but as Carl said, we've all been in that horrible situation, okay, where we have, it's either us or a family member, it's going to happen to everybody, where there's an incurable disease. Okay, and that's when we're all desperate. And that's when we're all going to say, wow, why didn't the world invest in this before? Okay, and one of the ways to get the world to invest in it is to make sure it's rewarded. It can't just be rewarded by these sort of value-based and cost of good arguments. And I really think that that's something that, as a society, we're going to have to come to terms with.
I always wish that George would really tell us what he thinks instead of holding back so much. She's so reserved. We're going to change gears now and invite Rachel up to tell us her story, which of course is the whole reason everybody on this stage is doing the work that they're doing. And if you don't mind coming to the podium, I think that would be great.
Hello, everyone. I'm Rachel, and I'm honored to be here with all the scientific minds to tell my patient perspective. Very much so a testimony specifically to Carl June and Bruce Levine's work at UPenn. But to give some background, my medical history story started back in 2008 when I was first diagnosed with acute lymphoblastic leukemia. I was 11 years old, and at the time, I went through a normal 2-year protocol full of chemotherapy. And as I like to say, my cancer responds well to chemotherapy, but my body does not. And so my immune system took a very strong hit, and I had a lot of small complications, including like appendicitis, pneumonia, things like that. But overall, was put into a full remission. And after 5 years of routine blood work showed a relapse, my first relapse. And at the time, I was a student at the College of Charleston, and I had to withdraw from school. And I think I got the phone call from my doctors on a Tuesday, and by that following Friday, I was already at home with a portacath installed, starting chemotherapy. And as many of you may know, the current protocol for a relapsed leukemia patient is another 2-year protocol of chemotherapy, much more intensive and invasive. So after 2 months of doing treatment again, I was at my lowest immune state, and I contracted a lot of bacterial infections and was put into the ICU when I actually had CRS before the CAR-T trial, which is ironic. With lots of bacterial infections, and was put into an induced coma and on an ECMO machine, which is a life-saving machine to take over the functions of my heart and lungs in order to try to fight off all this bacteria. Once I was on that machine for 10 days, and the doctors, they say you gotta see what your heart can do. And luckily, I'm very fortunate that my heart was able to recover and take over the function for my body again. And I continued my ICU stay with lots of physical therapy and occupational therapy. And after about 4 more months, there's no leukemia cells in my blood, and I made the personal decision to not continue with any chemotherapy due to the fact of how drastically it had affected my life previously. I returned to school, and about another year later, I had another routine blood work, and where the leukemia cells had returned. And at this point, I knew something had to be different. I couldn't just jump back into the chemotherapy world. My home team was very supportive of this decision, and they looked into many options, as well as me and my family, and we decided to enroll in the CAR-T trial at UPenn, directed by Dr. Carl June. And I had this specific cell that they'd been talking about, the C19 receptor, so I aligned perfect for it, as well as my prior knowledge with CRS, which is one of the main side effects of the CAR-T. It worked out really well, and I was able to slide into a position in the trial pretty efficiently and fast. My relapse was in October, and by the end of October, I was up at CHOP, where they did the first cell collection, and I went back home for about a month while they turned the cells into supercells, as we like to say, at the lab. And a month later, I was back up in Philly for a month and a half, where I had 2 infusions of the T cells, and had very minimal side effects, I'm fortunate to say. I was able to have very minimal hair loss, and overall stay outpatient the entire treatment, which was something very new to me. It's a huge benefit of CAR-T. And so that's the part that really gets me excited to talk about. The background of my health story is important to understand the difference in between what a gene therapy can do versus chemotherapy. And I'd just like to go ahead and thank all of you for here today and all of the science that is being made in the progress of CAR T and gene therapy. I'd like to thank all of you who have generously supported ACGT and the funding for many of these gene therapies that have been offered to me, and hopefully other patients along the way. And I just hope that the continued support will help make other patients be in a forever remission like myself.
My seat. So we're going to open it up to questions now. You sit in my seat. I'll just stand up here because I'm not answering any of these. Rachel, I mean, that was
So you never
incredible.
had
And
a
I think that, oh, are you
Never
mic'd?
had a-
Yeah.
Okay.
Here. Do you want to hand that to Rachel? I think we have to thank you for being brave enough to enter a clinical trial of an unproven medicine. I mean, you're the reason we have new medicines. You're so brave. What was that like? I know you said you were done with chemo, but what was that like thinking about taking a medicine that isn't on the market yet?
Well, yeah, it was scary to think about. For me personally, like I said, the possible benefits outweighed all the risk. I think, like Dr. June was saying, I'm one of those people that I'll take a risk when it comes to my life. I think more on a personal level, it was a hard decision to leave my home, I actually was there over Christmas, so we were 4 hours away from home during the holiday season, doing a treatment that we weren't sure was going to work, but had a lot of hope in it. So it was a difficult decision, but looking back, and even at the time, it was, like I said, the possible benefits really outweighed what the other option at the time.
Makes sense. Do we have questions from the audience? Yeah. So there's a mic coming to you.
Hi, my name is Kathleen. Should I stand up or?
Please.
I'll just stand.
Yeah.
Okay.
Yeah.
Okay. Hi.
Hey.
Hi. Thanks, Rachel. That was really great. I also have metastatic cancer right now, and I find out more results literally in 2 days, and I have some scans. So I'm involved in a malignant melanoma trial with. So you give me some inspiration because I chose not to do chemotherapy myself, and after I watched my mom kind of suffer for 4.5 years, so she passed away, and that's why I come here. It's April 14th is my parents' 1962 wedding anniversary, so my dad couldn't make it. He was too tired to come, so that's why we usually come together to these get-together galas. So anyway, that's why we usually come and have a wonderful time, and your speech was really inspirational. I wish you all the best, truly.
Thank you so much. You as well.
Thank you. Dr. June is awesome.
Yeah.
I follow his career like a Hollywood star.
As we should for scientists, right? I mean, this is one of George's main points.
Exactly. So I have a science question. So I have a PhD from NYU in carcinogenesis. So I understood actually what you guys were saying. In response to the cost of developing drugs, the only thing I would have to say is there some way that these you all with these big companies can make, I know, with a switch of a finger or a snap of a finger, drugs that are sold to the common person so that you can have like your gravy train of drug, so that you have a constant supply of like a aspirin type drug coming in, so that you can keep up with making these more expensive drugs for that are less of a price. I'm not a Wall Street guru. I never took banking or money management. I was too busy taking biochemistry as a major. But there's got to be some way of reducing the cost of these very expensive drugs. I know there has to be. As far as having your company have just like a major drug that everybody kind of needs and buys over the counter, if you will, so that the expensive drugs can be made with less amount of money. And I agree with you, sir, that to have people encouraged to be very smart and develop drugs, yes. Why not? Wall Street bankers get incentives, and I think scientists should be getting monetary incentives. That's wonderful. But I'm sure you guys can figure something out to make it cheaper for a common person to have access. My other question, but that was a comment. My other question is for the gentleman from I can't read that.
Select.
Andre.
Yes,
Yeah.
hi.
Actually,
So when you were gene editing, are you doing CRISPR now for gene editing? And then that's one part of it. And then number two, when you're editing the genes for the T cells, and then number two is, what actually were you targeting in the tumor? You said you were targeting for the tumor?
Okay.
Something in the tumor. What actually were you targeting?
Okay.
Or should I just read your paper?
No.
All right,
no,
let's start with
absolutely
the
not.
second part of your question with Andre, and then we'll address the pricing part.
And thank you.
Okay, I can answer for the target. So the product that we do actually are supposed to have, we're also already trying to work, and I think Community is also trying to work on trying to reduce the price here. But the initial objective of these drugs is not trying to reduce the price first, is to try to make them work. It's like the idea of being able to cure, for example, an acute myeloid leukemia is still a dream today, actually. It's like really a dream drug if we can do this, because you have like between 22 to 25,000 people that get AML every year. And like the death toll is like around half of these people die every year. So notwithstanding what the price will be, first let's try to make it work, and then also trying to work on this price. But the target is CD123. It's a kind of the same type of flag on the cells as CD19. And well, for now it's still experimental. We don't know how far this can go, but it's very encouraging. That's right. It's a unique on the it's unique on the compartment of cells. However, you cannot let the drug, as in your case, stay for the patient forever, for the simple reason that the progenitors of, for example, red blood cells, called the myeloid progenitors, express this flag, CD123, and therefore you will constantly wipe them off. And then the patient will have to live on a permanent transfusion, which is not doable really, or you'll have to wipe out the car at a time.
And on the CRISPR use talents,
Do we use CRISPR? So the gene editing, we do use CRISPR very regularly in our research labs, but the product that we're developing uses another different type of gene editing technology developed by University of Minnesota called TALEN. So you have like 4 different type of technologies called meganucleases can do this, zinc finger nucleases, TALENs, and of course CRISPR. So all this in an array of like gene editing
Let's
technology.
just make sure we have time just to spread it out, and we'll have cocktails where you can definitely follow up.
Follow up with CRISPR with him later.
Excellent. And we'll get back to the pricing question, but let's take
Community
another
needs
question from
CRISPR
the
right
audience.
away.
Just
Yeah.
very general, what's the outlook right now for NIH and federal government funding for your research?
I'll let my academic colleagues speak
Yeah,
to it
so,
first, and I'll give you my point of view.
You know, we have financial cycles, and unfortunately the government's funding of science has the same sort of sine waves. And we would, you know, a year-to-year budget is not the way to really make science planning. You know, unfortunately that's what we have. It's a political process. I mean, this year, the straw man budget was initially we were forecast to have the worst and largest cuts ever to the NIH in the initial budget, but it ended up there was a $2 billion plus up. So we had about 10 years constant decrements, you know, previously until the last year in funding in real dollars, from about 2005 down to just this last year. Then
When you say $2 billion plus up?
2 billion.
yeah, 2 billion.
What does that mean?
Well, the NCI budget's around 33 billion, and
The NIH
in
and the NCI.
NIH, yeah. So it's, you know, it's 2 billion out of 33 billion total, Now, and some of that's intramural, meaning it happens just in Bethesda, and then others, it goes to their universities around the country. The problem with cell and gene therapy is that, I mean, it had many years where it was disappointing. The results didn't work, and we could not get funding from the NIH to do our trials. So we had lots of basic science funding, and my lab could run off that, but the only way we, when we were ready with the CAR that now is FDA approved to treat humans, the NIH did not fund that. They said it's just, there's never been any cell therapy that worked and gene therapy. And the only foundations, and the only way we had it happen was philanthropy. And that was through, Barbara, the Netter Foundation, which was ACGT, we're here, and then also the Leukemia and Lymphoma Society. So foundations were more interested in this translational crossing the bridge with philanthropy, and we couldn't get, the standard government system to do that. It's a very different system in China. Now there, in 2010 when we had our first real strong results in leukemia, there was no trials in Japan or China, 0. And now they lead us. So they've come from, and they have done that because they have many more people than we do in universities, but they also have made it a government priority where they spend a higher percentage of GDP, much, much higher, on cell and gene therapy. They made that a national competitive priority.
Well, when
and
you
I've
have
talked
a
to that in the
president-
past, it was like a Sputnik.
The president is so concerned about losing ground to China. Has anybody made the point to him that we're losing ground in the medical race and maybe he should fund the NIH?
Yeah, so-
more?
Well, for example, for gene editing, it's spectacular.
Yeah.
I've been in this space for 30 years, and initially it was like few groups like United States, Europe that were working on gene editing. And for the past 5 years, you look, for example, you do PubMed, and then you have essentially Chinese papers that comes up in a row, and it's starting to be difficult. So like the balance starting to tilting in a very worrying way, I would say.
It's a tremendous threat to our whole economy in that, offshoring our IP. So it used to be at our universities, we would have brain drain, and we would harvest the best talent out of Europe and China and Asia. And now they get better offers back at home. So we train them here, they take back our IP basically and our training, and we don't have jobs because it's been of our prioritization at the national level. So we need to be very vocal that medical research, that's where innovation really comes. The pharmaceutical industry is really good at making things real and globalizing, but the innovation usually happens at academic centers. And they have just been really...
But just to clarify really quickly, you said this year was a surprising $2 billion plus up. So that's good, right?
Oh, it's great.
Okay, and is there reason to believe that trend is going to continue and will pass China, or do you think it was just a one-off, money fell down the well sort of?
So I think after many years, there's now a bipartisan effort to say medical research is important. And then we have our current administration, which is not really anticipated. So I can tell you, I have gone, and many of the scientists that I know, we've gone to Capitol Hill, and they now have, and in part it's because of people like Rachel. Before, we never had, living testimony like that. And that has made the difference, unfortunately. I can tell you that Emily Whitehead has been to Congress, and that has changed Mitch McConnell's mind. It's changed on both sides of the aisle. They now say these people vote, and it's really good if this research actually changes lifespan. They never saw that before. They saw, theoretical things. Now they see real palpably that medical research can really benefit and that cancer may be, Joe Biden was, when he saw what happened to his son, became, a real, apostle really to make the moonshot happen. If the election had turned out differently, then I would say we would have a $2 billion plus up every year for quite a while, and it would get up to close to what China does. But right now, it's a one-year thing. There's no long-term policy, and every year they have to go back at the NIH to Congress, and it's a separate line item authorization. And so it's a real problem. We need to have, come into Congress and say how important it is, and then because right now there's no policy that's a national policy, which is what it should be, both to bring this stuff across the finish line. And then in addition, it makes jobs here in the U.S. rather than making them offshore. And unfortunately, what Andre said is completely true. If you look at intellectual property filings, we are being drowned out by China now.
It's crazy.
It was they came out of nowhere, and they made a prioritization that they can do with the kind of government they have. And so part of the trade war thing is real. They have talked, in my own personal view, they've talked about the issues on IP and so on. There has been a lot of IP theft, but they're also investing in areas that make that it's a smart time to invest in.
But there's a really important sophistication and subtlety to all this. When people talk about the NIH budget, people have to understand if let's say it's around $30 billion, it's how it's divvied up. And what we have to understand, and Carl did a beautiful job of describing this, most of what led to this was really what they call basic research, not really translational research. Now, the problem with translational research is it's important to translate the basic research to patients and to save lives. But translational research tends to be much more expensive than basic research. And the beautiful symbiosis that we used to have in this country is that the NIH would fund the basic research, the most basic of research, and very occasionally fund translational research. And the industry would fund translation because the incentives were there to do that. If we shift and we start saying, okay, NIH, we're going to fund you, even if we can double the funding of the NIH. But if we put the onus on them to do the translation, we're actually going to have less basic research. And that still is the strength of the good old US of A. Because I'm sorry, I know the IP landscape better than anybody. The true IP is all coming from the United States. It's more on these translational things that other people are competing us with. If we destroy the NIH's competitiveness in basic research, that'll be the ultimate undoing of the whole system, because that's where everything comes from. And it'll also be the undoing of us as leaders across the world, because by and large, when you follow, like Carl did, the genesis of almost every new therapeutic in the world, it comes from NIH-funded research. So we have to be very careful. And it's very easy to convince congressmen or vice presidents, because they have a horrible tragedy in their family, to invest in the next treatment from a life-threatening disease on the translational front. It's much harder to say, invest in the Drosophila research, invest in the worm research, invest in that. And I think that we have to really understand and address this and be united. In that I think that NIH, we have to continue to fund the most basic of research, the things that lead to the CRISPR, which is then allowed to then be translated. And then we have to create the incentives for a symbiotic environment so then industry can then take over and put much more resources. Because as collectively, and we can't destroy this, the industry invests somewhere around $200 billion, which draws the NIH research in translation. We got to increase the NIH basic research investments, and we got to come up with independent funded vehicles, whether it's from the ACGT or at much higher scale from industry, to fill in the rest. And this symbiosis is important, and we have to understand it's not just the NIH budget. It's the NIH budget for basic research, and how do you pay for 10 times the cost of that in translation?
So I just want to pivot off that point, that you focus the question on the NIH for basic research, but the vacuum and the void is then left to companies like mine or Andre's or others, right, to fill that void from a translational medicine perspective. So you do that. But the other challenge that we're facing in the US is the incentives for companies to actually invest and do more. For example, I've got a number of colleagues that work in the orphan disease space. A lot of great work being done for smaller populations of unmet need, and nobody else doing it with these companies. The recent tax changes by the government really eviscerated them from an orphan tax credit perspective. And that was positioned as, hey, that's okay because the overall tax code is going to make it so much better for R&D innovation. That's frankly bullshit. It hasn't happened, and it won't happen. So the incentives for companies to really want to be in the US and really perform here is something that I would encourage all of us to think about and talk about a lot more. Because it's not just about NIH funding, it's also about keeping that whole ecosystem really attractive for innovation to continue. And that, for me, I see as a real risk in the next decade.
All right, we're running a
because
little bit
that is going to go elsewhere.
late, and I'm getting a little scared here. We've got time for two more questions. Does anybody else have a question that they'd like to ask before we? Yep.
Just following up on your statement on rare diseases, I'm involved in the Alpha One Foundation. And, we have about a half a dozen partnerships with biotech companies to bring forward gene therapy, gene silencing. It seems to me that the orphan drug community is going to be catching up, maybe surpassing what you guys are doing as far as science in the very near future. And secondly, part of that, you guys are trying to kill cells. We're trying to change the basic underlying mutation. Why aren't you looking at that?
So I welcome the orphan companies actually accelerating. You know why? Because we're going to cross-pollinate and learn from each other. So that's really important. And the second point is we actually are looking at cells, not just killing them. So, for example, within our company, we look at T cells as a whole. There are T cells that can be primed and made bionic, as Carl described, to kill. But there are cells that actually suppress the immune system, T regulatory cells. So we're also looking at therapies that we can create in the future through our research programs where we actually develop cells that don't destroy but actually dampen down the immune system as well. So we are looking at that. And there are many companies out there that are championing that in other diseases, not just cancer, but in the autoimmune disease space.
I think we had another question here, and we'll answer them all at cocktails, but we'll take our last question up here.
First, thanks to all of you, and thank you especially for sharing your story. It's very inspirational. A quick question for the entire panel. You've given us a peek at the future. It seems to me, based on your comments, that we may, as a patient population, need a new vocabulary with which to talk about our diseases and how they relate to your science, going beyond the organs of origin and getting into the molecular attributes of our cancers, creating new communities based on the targets rather than the originating organs. Can you talk a little bit about the conversations happening among basic scientists, translational researchers, and clinicians, including in pathology, to help educate patients and frontline clinical practitioners so that we can understand our relationship to the work that you're doing in a more effective way?
Great question.
So I mentioned, the trend of will the NIH accept or FDA accept basket trials and give approvals that way. I mean, science is changing in a way that it's very hard to keep up with, and for the lay public, it's impossible. This is so complex. Some of the ideas we're talking about. In my own area, we work in transfusion medicine, and the pathologists are literally being completely disrupted. I mean, it's going to be like going from barbarism, which surgery initially was, to, and it's going to be replaced. So it used to be microscopes were used to diagnose, and now it's all genomics and epigenetics. So that's how it is, and each tumor has a different path to become a cancer, and they all, that will then become different clubs of, say, I'm a KRAS tumor or not, or I'm a EGFR-driven tumor, and then there's certain targeted therapies. So people will be looking up on areas like Foundation Medicine, the sequencing of the tumors, and then finding the targeted agents. And none of us are trained to do that now. So we will have a big labor problem. I mean, pathology has to reinvent itself, and unless they go back to medical school, they're dinosaurs. I think the patients are having to keep up with this, and we don't even have the labor pool right now to do cell and gene therapy, because for many years people kind of snickered at it, and so you couldn't really there was no pharmaceutical industry, so there's really no job opportunity. You could do a postdoc at a university, and then say you wanted to work on CAR T cells. There was no jobs, and now we have this huge vapor. It's a vacuum because. So we have a labor shortage, and we have now to keep up with this information flow, so data handling is going to be central to how cancer is treated.
So I can give you an example of sort of the brainstorming and the dialogues we do within the company, right? So for example, Carl and I were talking to a colleague the other day about, the future vision, and that future may not be that far away, where you're in the OR and a patient is being treated on brain tumor stereotactically at that moment in time. Rapid real time, you're in the OR within hours, literally hours, we're going to get a sequence back, and we're able to work and deliver intratumorally, a universal CAR. And at the same time, we may be able to titrate with a switch technology to turn it off, turn it down, and modulate it. And in order for that to happen, you need to have that fertile dialogue happening in real time between multidisciplinary teams, right, with the industry. So that's kind of how we look at a patient profile and think of that ecosystem of thinking about what is possible here. And I don't think that narrative that I just described by Carl, I don't think it's that far away, right, when we think about what can be done. And I hope it happens in my lifetime.
Just to follow up on that comment, what kind of infrastructural changes will we need in order to provide the vast majority of patients with the kind of incredible genomic information I have, and perhaps you had, and a few of us very privileged patients have gotten? How can we provide all of that now, and how can we talk about it amongst us lay people and with our legislators and with our philanthropic friends?
I honestly don't know the answer to that complex question. I really don't. I think you go deep and do it really, really well with centers that really know what they're doing. And then, you have miracles like this, right, that do the talking for you and say, she's an example of a living drug. And she is. And I think that's what we need to happen. But in order for this to happen, it requires a real transformative step change in how we're thinking about translational medicine in this country and making it a reality.
I have a question for the assistant. How many people sequence their DNA in this system?
How many have you done it?
Only?
Shame.
Not even a 23andMe
23andMe
or something like this?
Do a spit
Okay, it's
sample.
like starting to be better, actually. So, like, the idea is that probably in the future, the medical file of people will be stored in the cloud with probably whole genome sequencing artificially.
Not on Facebook, right?
Cambridge Analytica.
Cambridge Analytica,
exactly.
exactly. With probably artificial intelligence being able to browse through this data and also connect through all the pathologies, their molecular profile, and also be able to sort out which kind of relevant data a physician at the hospital can sort out to exactly know what kind of therapy would respond or not. For example, for certain leukemia, you have poor cytogenetics that don't respond to certain chemotherapy, so no need to do chemotherapies at start. So maybe it's better to start with a CAR T therapy on the first line, etc. So this is probably the future of our, like in the 10 years, not that far.
Well, I feel like I have to comment on this because some of you may not know, Regeneron has the largest human sequencing effort going in the world, working with the best purveyors of electronic health records to try to create and build exactly what you're talking about, and create it in such a way so that medical practitioners and patients can actually understand all the imaging that you get, all the lab values that you get, plus your sequence information. And that is the dream. Right. But I have to say, people first have to build it. And it's going to take a while and a huge investment, which we're starting to make, to try to create these massive big data resources that put all the pieces of your electronic health record together, allow algorithms and so forth, software to make sense of it. And the hardest part of all is communicating in a way to even physicians, because as Carl said, right now most physicians are not educated to even deal with this, let alone patients. So this is the dream of the future. Many of us are making massive investments in order to do this. But it's going to take quite an investment and quite a bit of advances to do it. Cancer is certainly one of the places where the genome and the cancer can be put together right now. You're seeing examples of it. But to do it in a more holistic way for all diseases across everything is something that I do think as a healthcare system we're going to have to solve, because that's one of the ways we're going to get the efficiencies we're going to need to be able to deal with our healthcare load and burden of the future. But it's going to require a lot of investment and a lot of work by a lot of people to turn it into reality, but it is the dream.
I know we have more questions. We're going to have to hold because as a TV reporter, I break out in hives when I don't follow time cues, and we're already over time. So we'll go to cocktails, but we have to thank George, Oz, Andre, Carl, and of course Rachel for this incredible discussion.